Hello, hello. A very good morning, all of you. Today I am having a privilege to introduce one of my gurus, starting from my MSc days. Actually, I was working in his lab uh, almost a big part of my MSc, and then from that time onwards, whenever I face any problem, I always consulted him, and till now he is giving a lot of suggestion for improvement of my own research work. So he is Dr. Sunil Mukherjee, renowned plant biologist. He earned his BSc degree in 1969 from Calcutta University, MSc in physics. So you can able to see that he is basically a physics person and how he has transformed himself to a molecular biologist. So he did his MSc from Vishwabharati University in 1974. Post-MSc program, he started on theoretical nuclear physics from Shah Institute of Nuclear Physics in 1976. And then he did his PhD degree in 1983 from, again, Calcutta University on biophysics under the supervision of Professor R.K. Poddar. He joined biochemistry department of Johns Hopkins Institute, University, USA as postdoctoral assistant during 1983 to 84, and later worked in the molecular microbiology department of Duke University, USA for 6 years as a research associate. He returned to International Center for Genetic Engineering and Biotechnology, ICGEB, New Delhi, in the Plant Molecular Biology Laboratory in 1990. He was a visiting assistant professor at the University of California, Irvine, USA, 1992, and a visiting professor at the CNRS, France, in 2002. Dr. Mukherjee has been working in the field of DNA replication since 1984. At ICGEB, he started working on P. chloroplast DNA replication and established an in vitro system for replication. Later, his interest shifted to genome replication of plant DNA viruses, especially the Gemini viruses. He established a yeast model for Gemini viral DNA replication, the exploitation of which led to the identification of important host as well as the viral factors necessary for viral DNA replication. He is an internationally recognized researcher in the field of RNAi technology. He developed tomato transgenics that expresses artificial microRNAs which consequently silence the critical genes and of tomato leaf curl viruses. He has guided 8 students in their PhD program. Professor Mukherjee served as a member of different expert committees of national science funding agencies, DBT, and all other science forums. After his superannuation, he was awarded as NASI senior scientist and worked at University of Delhi. And presently, he is INSA senior scientist, and he is working in our division, Division of Plant Pathology, IRI. He is the recipient of fellow of all the academies of India, National Academies of India, and he also received many awards. So I am very small in front of him to tell about him. So now I'd like to hand over the mic to him to tell you about his experience on he has worked on RNAi technology for long. So in RNAi technology, what is happening, and genome editing, how it is different from that. So he will compare and tell you about this. Thank you. Sir, over to you, sir.
Yeah. Okay. So thanks, Anirban, for the kind introduction. In fact, the introduction is larger than the talk I would be giving. And thanks to the organizers for giving me this invitation. Let me start. See, I have quoted something from a journal, FUBS journal, and they say what's happening in genome editing stuff. You can have designer babies, okay? And this is not a theory, that has been done already from China. From designer baby to second green revolution. So you can have rice seeds with improved yield. The plant which can be tolerant of all kind of stresses, biotic and abiotic, to have a second green revolution. All these things you can do with genome editing. So it says there is nothing that genome editing cannot do. And this technology is far more superior to any other technology which has been invented before. So such is the power of CRISPR-Cas technology. And on the top, there is a cartoon which says how to create mouse with knockout in various genes. Okay, you take the embryo and then deliver your CRISPR-Cas reagents, basically hgRNA and Cas9. And after some division here, you transplant them to the ovary of the mouse, mother mouse, and the litters come out, and the litters will have the phenotype for which you did the genome editing. In this case, particular case, the skin color is noted. In this way, you have a mouse library with all kinds of knockouts. There are many different companies that have come out till now. So this is the silencing technology. And previous to that, there was another, not previous to that, I mean, it is still continuing. Another technology which is known as knockdown technology. This is knockout, and the knockdown technology. And for knockdown technology, there is antisense technology, there is RNAi technology, which we will be talking about today in brief. So I have been asked by the organizer to give a lecture comparing this technology versus this technology, that is knockdown technology versus knockout technology. And in my opinion, that's actually, stupendous task. There's so much of literature, so much of information generated that in fact I was initially at a loss how to proceed. Then I thought, well, I'll make something. So whatever I'll be telling here is not the complete story. It's just impossible to compare all the comparative phenomena. But I'll give you a hints in how one is different from the other, how one is effective in one case, how one is defective in the other case. Okay? And if you have any question meanwhile, please raise your hand for clarification. Okay. So both technologies are silencing technologies. So what does it do? They are important for causing loss of gene functions. And knockdown causes loss working at the mRNA level, whereas that's the basic difference, and the knockout works at the DNA level. Okay? And knockdown is the process that can handle, this is important, that eukaryotic transcripts, eukaryotic mRNA. Prokaryotic mRNA cannot be handled by this technique because the prokaryotic cells are RNAi incompetent. To be RNAi competent, you need a lot of RNAi factors. Then the factors are dependent on the system, like in Arabidopsis plant, there are about 200 different RNAi factors. Similarly, in different systems, there are different factors, but the prokaryotic cells do not harbor these factors at all. So this process cannot be done in the prokaryotic system, whereas the knockout started with the prokaryotic factors. It's essentially prokaryotic material. To defend the prokaryotic cell against bacteriophages, consecutive plasmids, and any invasive plasmid. But then later on, the system behind, the proteins behind, and the RNA behind it was transplanted into eukaryotic system so that you can get knockout in the eukaryotic system. And this system works on DNA, and this system works on mRNA while keeping the DNA intact. I thought I will be talking to you about the RNAi process because the editing process, the knockout process has already been talked to you. Okay? And let me start with the mechanism of RNAi process, how it happens. This is a whole cell, and this is the nuclear part of it, and that's the cytosolic part of it. Okay. In the nucleus, let's say viral DNA has come in, or you have introduced a gene, transgene, in the chromosome. Okay. So this will transcribe because this has all the transcription facilities in it. So an mRNA will come out, and this mRNA, in the case of RNAi, this mRNA will be subjected to cellular RNA-dependent RNA polymerases so that an mRNA is transformed to a double-stranded RNA, and that travels to the cytosol. There are mechanisms within the cell. Once this double-stranded RNA is done, the cell goes berserk, because cell cannot handle this double-stranded RNA at all. So the RNAi factors or the surveillance factors come in that case. A set of complex proteins called dicer complex, of which dicer is the main component, act on this double-stranded RNA. In fact, each dicer has its own cognate RNA-dependent protein, RNA binding protein. So this dicer, along with RNA binding protein, recognize this double-stranded RNA and literally cuts them into species. Okay? And these species are of definite size, definite chemistry. Like, it has protruding ends, 3' protruding ends, 5' phosphate, 3' hydroxyl end, etc., etc. And it has a definite size. And the size depends on the characteristics of dicer. The size could be 21 mer, 22 mer, or 24 mer. Okay? These are the three options the dicer can give. And once the small RNA comes up from this double-stranded RNA, this is subjected to the interaction of many different proteins, at least a set of 7 different proteins, 7 to 8 different proteins. And those are called risk factors, RISC, risk factors. Okay? Once the risk factors, especially the arginate component of it, attaches to it, out of these 2 strands, 1 strand vanishes. How it vanishes, that's a bit debatable. I'm not going into that. But 1 strand vanishes, but the antisense strand remains with the Rix complex. And then the Rix complex becomes a site-dependent RNAs. Okay? So why site-dependent? Because there is an antisense component. It can recognize its cognate mRNA by Watson-Crick pairing. And since the protein arginate is present here, it makes a cut in the middle of this local double-stranded RNA, right in the middle. Okay, so the mRNA, you see the mRNA is getting degraded, and once it is degraded, then the exonuclease factors of the cell chew it up to nucleotides. So this mRNA is rendered completely unfunctional, non-functional, because it cannot make any protein and what does. In some cases, you will find that this mRNA is not cut, but these protein factors are stuck to this mRNA. And since they are stuck, this mRNA cannot make any protein. So that is also a non-functionality. So this is called a phenomena called post-transcriptional gene silencing, because mRNA is made and then mRNA is destroyed. So gene silencing, but it is post-transcriptional. Sometimes it happens the small RNA, siRNA, travels back to the nucleus, okay, to the DNA, and makes a complex here so that the DNA becomes transcription incompetent. It cannot make an mRNA. And that is called also a silencing, but that is called transcriptional gene silencing. Okay? So this is essentially an overview of RNAi processes. Any question? Is everybody with me? Yes, please, go ahead.
As you said that after nuclear activity by RNA, this complex, there is the chopping down of mRNA.
Right.
So is it also some kind of knockout?
This is not knockout. Knockout will be when the DNA is affected, here the mRNA is affected. Okay? And you see, in the cell, there is not one mRNA. All right? There are many mRNAs. And similarly, it is quite possible that all the mRNAs are not affected, because siRNA cannot handle that many. And that's why this process is not a complete loss of function. Most of the time it is incomplete. But in some cases it becomes complete if the concentration of siRNA is very, very high, so that it can handle all the mRNA. In that case, this loss of function will be like a null mutant function. Okay? But in the majority of the cases, since the mRNA concentration is higher than the siRNA, this loss is not a complete loss. It is a partial loss. On what factor? Okay. That's a good question. So this is an siRNA, okay? And siRNA does its job, and then it gets released so that it can act further on another mRNA. So in that case, it is catalytic. But even if it is catalytic, the mRNA concentration could be very, very high. So if there is a process by which this siRNA could be amplified, okay, then its concentration will be very high. And this amplification happens because of the presence of the cellular RNA-dependent RNA polymerase, which is present in plant fungus, not in animals. In some cases, some insects have it, but like we undergo RNA processes, but that siRNA cannot be amplified in our system because we lack RNA-dependent RNA polymerase. Okay. So the question is, how effective is this RNAi? I just mentioned that that the siRNAs could be amplified in some system, okay? And if they can be amplified, then they work very well, okay? Then the loss in that case, loss of gene function, will be like a null mutant. For example, in nematode, there is a muscle protein called UNC22. Okay, when this process was discovered first in the nematode, then the UNC protein was targeted, and that targeting led to a Nobel Prize. Okay, this UNC protein is such that when you introduce, at that time siRNA was not introduced, a long piece of double-stranded RNA was introduced from outside, okay, and the double-stranded RNA of UNC22 completely knocked off all the UNC22 proteins. So the nematode was defective in muscle activity, and that act like a null mutant. So here the loss of gene function was complete. Complete because the UNC22 RNA was amplified. The double-stranded RNA was amplified within the system. It is not only complete, the animal which was treated with double-stranded RNA, its offspring were also defective in that muscle function. So it behaved like a systemic function. But the point is that most of the time it doesn't happen. That you should keep in mind. The discoverer was fortunate that they started with the UNC22 protein. Had they started with another protein, maybe the life would have been completely different. There would have been no Nobel Prize. The other point of constraint is that RNAi has a lot of off-target effect. Okay, what do I mean by off-target effect? Like you have designed, let's say, an siRNA to destroy one gene. siRNA has a specific sequence, and it will work very fine. When it works, the on-targeting effectivity is 100%. But the cellular chromosome is very, very big, okay, and that transcribes lot of mRNA. Like say an organism like us, plant, okay, mouse, etc. They have effectively 30,000 different genes. It could be 30 to 40,000. And each gene, the mRNA of each gene, on the average codes for say 1,000 nucleotides. Then length of the mRNA is 1,000 nucleotides on the average. And from each gene, there are different spliced versions. On the average, 6 spliced versions are made. So the mRNA target for an organism coding for 30,000 gene will be 30,000 plus 1,000 multiplied by 6. So this is the target of the mRNA. Now, one thing you have to keep in mind, you have introduced a 20 nucleotide siRNA, all right? And the functional characteristics of this siRNA is not 20. If you have seed sequence that is starting from the 5' end, only 7 nucleotides, that 7 nucleotides will be suffice to cause a knockdown. All right, and if if this is the target, then you can easily calculate how many different targets are possible. If you have, let's say, a stretch of 7 nucleotides, what is the possibility that the next 7 nucleotides will have the same sequence? How is that calculated? The possibility is 4 to the power minus 7. All right. So, if we travel a distance of 4 to the power 7 nucleotides, which is about 16,000 nucleotides, then you might find another same sequence. And your target is now this big. So, how many targets you can get? This divided by 4 to the power 7, which will be more than 10,000. So, you have introduced one siRNA which has one on target, but it has more than 10,000 off targets. All right. So, it has a lot of off target activity, and that's what the undesirable part of it. Is this part clear? Yes.
I'm working with a construct where I'm introducing a double-stranded RNA. So I expect it to get cleaved into siRNAs once it is into the system. However, I don't know what are the possible outcomes of that cleaving of double-stranded RNA. So this is the number of off targets for one siRNA. So
Yes.
given I have a 340 basis of double-stranded RNA, so how many more off targets will be there?
Yes, there will be lots of off target, but the important question is how effective is the off target? Will that give rise to a different phenotype? All right, your on target is 100% loss, but here for all off targets, the loss is not 100%. It is varying. Okay, in some cases, maybe 10%, some cases 20% loss, etc., and etc. So the important question is, will the off target give rise to any phenotype? If this gives a different phenotype, then that's a problem. If it doesn't give a phenotype, then it's an acceptable thing.
What is the length of the siRNA that you have designed?
Anywhere between 20 to 24.
And if my gene is very large, like around 1,000 something.
Sure, sure.
What portion should I target?
Okay, there are actually web programs which will tell you which side is the best, but no web program is perfect, because nobody knows at which side of the mRNA proteins will be occupying. It's very difficult to predict. So what you have to do, not to design one single mRNA, non-single siRNA. You have to design at least 3 siRNA from 3 different locations of the same gene. Okay. And for this kind of knockdown, one good thing is that it is very easy to operate. That is, you have to give only siRNA, one siRNA or 2 siRNA from outside. And introduction of the siRNA within the cell is not difficult. There are sometimes cellular receptors which accept the siRNA, and on the top of that, there are many surfacting agents, okay, which will help the siRNA to get into the cell. So, as far as operational part is concerned, knockdown in that sense is very easy. This already you have learned, I guess, right? How to have knockout in the prokaryotic system, because it is essentially a prokaryotic system. if you look at the chromosome, you will find a cluster repeats. There are repeats followed by spacers, right? This all you have learned. And what is the function of the spacers?
It kind of acts as an immunity because the next time it is invaded by the
Yeah. Either bacteriophage or consecutive plasmid or any other invasive plasmid. Then the spacer sequence will have that information. And along with the spacers, there are CRISPR associated proteins. Okay? Depending on the bacteria, you will find different types of CRISPR associated proteins. The most proteins, in most of the events, the Cas protein which is utilized for biotechnological work is Cas9. So far, 16 different Cas proteins are reported. Okay? And out of them, Cas1 and Cas2 are kind of invariant. Like if a bacteriophage comes, the part of the DNA will be inserted into these repeats. Okay? And Cas1 and Cas2 protein does this job. So Cas1 and Cas2, all kind of system will have Cas1 and Cas2. And for the others, no, it depends on system. And there are actually many different 16 types of Cas, but they can be divided into two separate classes. One class is like Cas9, and the other, because Cas9 is alone and it can alone do the immunity job. Okay? And for others, for immunity job, you need a combination of proteins, and those are called class 2 proteins. And how does Cas9 work? That must have been described to you, right? All of you are aware of it. Okay, so because of that activity, the Cas9 cuts the invading phage, the DNA of it is cut, and that's how bacteriophage is completely destroyed. So this is the prokaryotic scenario. The question is, can we apply it to an eukaryotic cell? The answer is yes, but in that case, things will be a bit different. If you insert Cas9 in an eukaryotic cell, you will get cut. We have described before. The DNA will be cut, and after this cutting, there will be a lot of repair enzymes present in the eukaryotic cell, which are not present in the prokaryotic cells. And those repair enzymes, mostly Ku type of proteins, will attach here. Okay? And they will try to bring these two broken parts together. And during that process of repair, at this area, either there will be deletions or additions. It's called indel. Okay? Del is deletion and N is integration. There will be addition or deletion so that the reading frame will be disturbed. Because of these mutations, the reading frame will be disturbed. Pretty soon, there will be a stop codon inserted. In that way, you are making mutants. Okay? This knockout process is giving rise to mutation in the gene. But sometimes the mutation is such that the reading frame is maintained. You get mutation, you get addition or deletion of few amino acids, but the reading frame is maintained. So those will be escape mutants. They might have biological function, they might not have. and I am sure that when Cas9 was discussed to you, HgRNA was talked about, right? You know all about HgRNA. Fine, so I'm not going into it. Again, the same question, how effective is the how effective is this knockout system? Okay. Once the DNA is cut, obviously that's a complete loss. No mRNA, no protein, nothing. So in that way, it's very good. The second question is, does it have any off-target? The answer is yes, it has also off-target, but the amount of off-target or the number of off-targets are much less compared to the knockdown system. Okay? If you remember that with 30,000 genes, we had at least how many? More than 10,000. Okay? But one thing you should keep in mind, the estimates that we are talking of is simply a theoretical. Okay? In reality, it could be much higher than that or much lower than that, because it all depends on the sequence. So I'm just trying to impress on you that the off-target effects of knockdown phenomena is much higher than the knockouts. That's the point I'm trying to make. And for that, I'm giving you some statistical illustration. But this statistical illustration may be far away from the reality. Okay, how to calculate the off-target here? Again, we take a system which can encode 30,000 genes, okay? And this 30,000 genes will be in the eukaryotic portion. But there is also a heterochromatic portion of it, okay? And the normally heterochromatic portion is much larger than the euchromatin. So what is the DNA size of human being? Or any mammals or plant? What is the DNA size? Sorry? 3, not MB. Billion, not million. Okay? So it's about 10 to the power 12. And if I take that, 10% of it is euchromatin, then the gene which you are encoding is about 10 to the power 11. And you are making a knockout in one gene on target. So will you have any off target? So the HgRNA has the catalytic part of 20 nucleotides. Rest are matrix, right? That's how HgRNA is defined. So within this 20 nucleotide, again, it can accept mismatch of at best 4 base pair. So the catalytic part of it becomes 16 base pair. So what is the off target? If you have 10 to the power 11 target site, then 10 to the power 11 divided by 4 to the power 16 gives off target for 4 mismatches. For 3 mismatches, 10 to the power 11 by 4 to the power 17. For 2 mismatches, 10 to the power 11 divided by 4 to the power 18. So that comes out to be an off-target number of 32. In the case of knockdown, it was more than 10,000. In the case of knockout, it is in the ballpark of 30. So it has much less off-target. Okay? Again, I'm reminding you that this is just a statistical calculation. Okay? This is just to impress on you that the off target for knockdowns, for knockouts, are much less than the knockdown processes. Is that clear? Okay. But it has difficulty also. Like if you want to introduce this system, you have to introduce the sgRNA, you have to introduce also the Cas9. So RNA and Cas9. So introduction of Cas9 is generally done by agrobacterial route. But in many plants, agrobacteria cannot deliver the cargo it carries. So in that case, virus vectors are only effective, but virus vectors can easily deliver sgRNA, but they have difficulty in delivering proteins like Cas9 because the cargo is just too big. So operationally, it is much more difficult than knockdown processes. Okay, there are some examples that what is this? Can you recognize? Just liver, and the liver has. There are many diseases which initiate in liver, but though it initiates in liver, now it affects the whole system. So in the liver, now there is liver-specific delivery system available. Okay, and that's how liver-specific siRNA are very, very effective. Knockdown can be achieved here, and at the same time, there are some liver cells in which you can do the knockouts. Like over here, a disease which is induced by the virus HBV. Okay, and HBV can cause all kind of ultimately liver cancer. People can die. But to prevent that, knockout could be effective. There is a gene called APOE. If you knock that gene out, then the virus cannot survive. So this has been achieved. So here we can do both knockdown and knockouts, and obviously knockout is much easier, more effective. And as far as blindness is concerned, that is one area where knockout has done kind of miracle. There is a gene called, what is the gene? MARC3. MARC3 mutation caused blindness from the birth. And I told you earlier that mouse knockout libraries are available. Is almost in each gene, the knockouts are available. So using those library, people have found out that for blindness, 347 genes are responsible. And out of these 347, 86 genes were hinted earlier by knockdown system. But now we have additional 261. And these are all possible because of application of knockout systems. So loss of function by knockout and knockdown, in most of the cases, parallels. Okay, they have the similar outcomes, but the degree is different. Knockdown is much less effective than knockouts. Like there are many invertebrates in which knockdowns and knockouts have been applied, starting from nematode, Drosophila, many different invertebrate systems. Both have been applied with similar results. Similarly, for vertebrate, zebrafish being the top one, this system have been applied with many parallel results. But there are cases where the outcomes of knockdown is different from the outcome of knockouts. Okay, that's where that's how it is interesting to compare. And I wanted to compile all the discrepant phenomena together and found I couldn't include all, but I found that they could be classified in 4 different classes, okay, where knockdown phenomena outcome is different from the knockout. 4 different classes. The first class that I'd like to describe to you is because of phenomena called genetic compensation. And genetic compensation happens in yeast, fungus, nematode, almost all organisms. What is genetic compensation? If you knock out a gene, you may not get any phenotype at all. Okay? It's because once you knock out a gene, another similar gene which is normally cryptic can come up, can compensate that effect of the gene. So there won't be any phenotype at all. Although your knockout process is right, everything is right, but you don't get any phenotype. But you will get the phenotype if you apply knockdown processes. Okay. In zebrafish, there is a gene called epidermal growth factor-like protein 7. Okay, and this stays not only in epidermis, but for the vasculature system, like through which the blood can supply, is affected. So when you knock out this gene, another gene, which is what is it called? Amylin 3A gene expresses. Okay, so this gene compensate for this gene, so you don't get any phenotype in the case of knockout. But in the case of knockdown, you get phenotype. Okay. In the case, say I am a plant person, I work in plant, and in the case of Arabidopsis, similarly there is a gene, auxin binding gene. If you knock it out, there is no phenotype. If you knock down, there is a phenotype. And I would like to describe some experimental results for this ABP1 gene. Am I making clear what I mean by genetic compensation? Okay. ABP1 gene structure I am showing here. This is the 5' untranslated region. This is the 3' untranslated region, and the green ones are the exons. Intervening black ones are the introns. Okay. Now, which area to target? for knockdown, for knockout? Exons. But which sites? If I use Cas9, in order Cas9 to be effective, you need a sequence motif in the DNA. What is it? Very good, very good. And NGG is the PAM motif, right? So in this case, this is the motif. So this site which can be targeted is about 20 nucleotides from here. Okay? And so you make, this is the sequence, 20 nucleotide, then you add sgRNA and etc. So your sgRNA will be made. And sgRNA you transcribe using this promoter, 35S promoter. Okay, so that's the sgRNA component of it. And Cas9 protein in this particular case, you wanted the protein to hang on to the nucleus. So you add nuclear localizing signals. So this protein is then expressed in the nuclei. Now once it is expressed, then the pre-mRNA, okay, the pre-mRNA will be targeted right at this site. All right, the pre-mRNA come out, and the DNA here will be targeted at this site. And then if you want to know whether ABP1 protein is targeted and it has vanished from the system or not, then you do a technique which is called Western blot. All right, you see, when you did the knockout, there is no protein. So ABP, using this system, ABP1 protein has been knocked out completely. And whether the targeting is right or wrong, then you do some sequencing around this region. And if you do the sequencing, you see there is a deletion in this part. All right, so the knockdown is not only functionally complete, structurally complete. So you have done a knockout of ABP1 protein. The question is, what is the phenotype? This part is the wild type, and this is the roots, okay, and this is the knockout phenotype's root, and the root structure is completely different, not different at all, the same. And then you look at the leaves, okay, in the seedling stage, this is in the seedling stage, and there is no difference. Middle stage, there is no difference. Mature stage, there is no difference. So ABP1, if ABP1 is defective, then its phenotype will be shown in the length of the plant leaves, okay, and the shape of the plant leaves. But here, absolutely, there is no difference. Why there is no difference? And why there is no difference? There is a big debate about it, and that's why I didn't put up the slides. There are some speculations that ABP2 protein comes up. ABP2 gene compensate for ABP1. Okay. Let's see what happens when you do the knockdown. Here the knockdown was done by antisense. In the system, the antisense RNA was introduced, and again, if you do a Western blot, you know, in the wild type it shows up, but in the plant which contains the antisense transgene, there is no protein. Okay, and if you look at the seedling structure, this is the wild type seedling, and the wild type seedling looks very different from the knockdown seedling. Okay, and let's look at the leaf. In the case of wild type leaf, you look at this forget this part, look at this structure. This is a small leaf, and then when it gets bigger, it is like this. But when you look at the knockdown, you see the difference. Because ABP1 protein is not there. And that's how leaf size is small, and it looks very different from the wild type. So here knockdown works, but not the knockout, because of genetic compensation. It's clear? Yeah, please. Because it is compensated by another gene.
But why it is compensating? Like due to no editing of other genes focus ABP2 in this case, that's why it is expressing, or there might be that ABP1.
See, right. The point is why the genetic compensation is happening. Again, there is a debate in the literature. Whatever you hinted is of off-target effect, right? That could be one of the causes. The other cause will be, like say, in the case of zebrafish, in the case of zebrafish, the epidermal growth factor like protein 7, that acts as a repressor of the gene. Okay, and when that gene is out, when that protein is out, what is the other name of the protein? Amylin. Amylin protein gets expressed because its repressor is out. So how the genetic compensation is done, that is a bit debatable. But the phenomena is that we see genetic compensation. Is that part clear? Mechanism part is not known, but in the case of epidermal growth like factor 7, that mechanism is known. Okay, but for ABP2, it could be different. For the third gene, it is different. So we don't know what really it causes, but we know this happens. Yes, please. Knockout. In the case of knockout, there is no genetic compensation, but we see the phenotype in the case of knockdown. Please. Redundancy, see, I won't like to call redundancy, okay? Because redundant genes will have almost same sequence, whereas here the sequences are widely different. Now this is not receptor, this is actually a negative feedback loop. Okay. So one acts as a repressor. When the repressor is out, the gene is expressed. So this is negative feedback loop. So one class I have described. Now I am describing another class. Okay. And this happens mostly because of off-target effects of knockdown. And this is zebrafish larvae. Okay, that's how it looks. And there is a gene is RHC glycoprotein B. Okay, this is the gene which causes ammonia secretion from the skin of zebrafish. Now, you can cause knockout, you can cause knockdown, both. In the case of knockout, the phenotype is almost like zebrafish, but its tail is a bit distorted in the case of knockout. And in the case of knockdown, depending on the dose of knockdown, here the knockdown was made by antisense RNA, and a special type of antisense RNA which is called morpholino oligonucleotides, because they are very stable, and then then the exonuclease cannot degrade them too much and all those things. So the morpholino oligonucleotide was used for knockdown, and depending on the dose, you see how the tail gets distorted. Okay, the more the dose you give, more is the distortion, and it happens just because an off-target effects. Why it is off-target? Okay, this is the knockout phenomena, and in the knockout phenomena, if you keep on adding this morpholino, then you get the same phenotype. Okay, so this is just an off-target effect, huge off-target effect, and the off-target effect is giving rise to a new phenotype. Okay, here it is now completely evident that off-target is doing something which is give rise to a phenomenon, now a phenotype. In many cases, it may not give rise to a phenotype, but here it is giving a clear phenotype. So, knockdown with this gene now is not desirable, because an off-target effect, and that off-target effect has a clean phenotype. In fact, I have a friend who works in the field of zebrafish, and she doesn't want to believe any result which comes out of the knockdown treatment. She says, I don't just believe, because the interpretation is too difficult to do. You don't know whether the gene is knocked out, or the gene, the phenotype that you are seeing is because of off-targeting. Her opinion is that if you have a null mutant, okay, then in the null mutant background, if you do the knockdown experiments and you don't see any results in addition to the null mutant phenomena, then it is believable. Otherwise not. So there are extreme views are there in the literature also. Okay, this is just simply a measurement of the the bending that you have seen just a few minutes ago. This I can also skip. Then there is another class, third class, but it is really not third class, it is superior class. Here, the protein are also important. Okay, the knockdown and knockout will be different depending on the protein that you are targeting. Okay, there could be some proteins which have very, very superior catalytic activity. What it means that if you knock down, let's say 90% of it, but still 10% is remaining. But that 10% is good enough to keep the normal function. There are many examples like that. For example, the phenomena epilepsy. Okay, epilepsy, how it happens? Okay, there is a protein called antiquitine, which is encoded by this gene, aldehyde dehydrogenase. And antiquitine lowers the level of aldehyde, is amino dipic semialdehyde. And this aldehyde, in its turn, degrades a chemical which is called pyridoxine. And this pyridoxine, what it does, in the brain, it degrades amino acids like lysine and neurotransmitters. If lysine is present and some amount of neurotransmitter is present in the brain, then you get epilepsy. But if you say knock it off, okay, or knock it down by 80% even, then epilepsy may not happen at all. In order epilepsy to happen, in order epilepsy to not to happen, you need to knock down this gene completely. Knock out this gene completely. A loss of function of 80% may not be effective at all. So here you see the power of knockout versus knockdown. Knockout is actually effective, but not the knockdown, even if the knockdown is effective to the percent of to 90%, because you are handling a protein which has superior catalytic activity. So this is another class of protein. And lastly, I won't say last because there are many other different genes that I have not handled. Some genes which are indispensable. So if you knock that gene off or knock the gene out completely, then that's a lethal phenotype. The organism cannot survive. So knockdown in that case is lethal. You cannot do the knockout experiment at all. But since knockdown is partial and not permanent, okay, you can do knockdown experiment on those genes and get the phenotype out. So here, knockdown is a bit more effective than knockout. And besides this, there are many organism-specific phenomena. Like, if you take zebrafish, the important thing happens that, mother gives lot of mRNA to its offspring, okay? And what is the result of that? Let's say you have taken a father zebrafish and a mother zebrafish where both are knockout in one allele. So this is heterozygote. Heterozygote mother, heterozygote father. If you cross them, then you get the descendant which will be homozygote knockout in that particular gene. It's quite possible. So you would expect that that gene function will be lost in the homozygote, but it may not happen because the heterozygote mother have passed the mRNA, wild type mRNA, into this homozygote. So in the early period of development, you may not see any effect at all with this homozygote knockout. All right, but if you do the knockdown, The knockdown won't spare any RNA, any mRNA, whether it is coming from organism itself or from the maternal. So knockdown will be effective. Okay, so this knockdown versus knockout phenomena, some organism-specific phenomena you might see. And there are other phenomena also, like digital versus analog. Okay. Let's say you get loss of function of 50% by knockdown and same loss of function by knockout. What's a disease like Huntington disease, it happens in the brain. So if you do a 50%, if you do a treatment which causes 50% effect down, then if it is done through knockdown, then all the cells which are nearby will be affected similarly, homogeneously, because blood will come, they will deliver the knockdown reagents, and let's say 50% knockdown has happened in all the cells. But if you do a knockout, then there are three possibilities. Either both the alleles are affected, complete knockout, or only one allele is affected, 50% knockout, and nothing happens. So there are three ranges only. And if you, within the ambit of 50% loss, you can get some cells where there is complete reversal, complete knockdown, complete knockout. So this small amount of cells which have undergone complete knockout, they might cause a simple reversion of the disease. Okay? So here knockdown is not effective, but knockout is effective. Similar kind of phenomena you can also see. So okay, so this is another important point while you discuss things between knockout and knockdown. Knockdown phenomena, okay, the research of it started around this period, 1990. And that's when RNAi phenomena in the plant was discovered. Do you remember what was the phenomena? The petunia. Okay, the petunia study began with this in the 1990. But knockout phenomena started revealing from 2011 or after. So knockdown research has a head start of about 20 years. So we have known a lot of literature, lot of things about knockdown. But knockout in that sense is a pretty young research thing. And a lot of things we don't know yet about knockout, the proteins that causes the knockout. Okay, I told you earlier that about 16 different Cas proteins are known. Still, I'm absolutely sure more will come. And for each Cas protein, there are subspecies. Like if you take Cas13, there are A, B, C, D, 4 different subspecies. So in this way, a lot of discovery is being made in the Cas field, that is in the knockout field. But as far as knockdown phenomena is concerned, a lot of things we know. Still something is emerging but a lot of things we know. So, manipulation of knockdown can be done in very different manners, but not knockout. Okay? This is one part, and the other part is that as far as knockout is concerned, there is only, if you take Cas9, is only one protein. And this protein, you can do a lot of different types of engineering to get different outcome of knockout. Like there are two nucleases of Cas9, you can make them defective, and this defective Cas9 can bind to the DNA with equally strong affinity, but it cannot cut DNA. Now, what is the effect? Effect is that if you design a defective Cas9, then you can stop replication, you can stop transcription at the site you require. So, and also there are many different types of Cas9, like I told you the Cas9 has some off-target effects. A careful design of Cas9 can reduce the off-target effects. But for knockdown proteins, you cannot do anything. You cannot do anything because there are lots of knockdown phenomena there, and if you tamper one protein, that may not be effective at all, because knockdown is a concerted phenomena of many different proteins. So engineering of knockdown is very difficult, but engineering of knockout is very much possible, and you can get desired results. You can get different catalytic activity, you can get less off-target effects, and so on. And Cas9 has a PAM motif also. You can design Cas9 so that PAM motif is relaxed. It is not NGG, but it will be NNG. Similar things you can already design. So, in that case, in that sense, knockout, you can handle it in many different manners, but not the knockdown. But the delivery part, is, as far as knockout is concerned, delivery is a bit difficult. As far as knockdown is concerned, delivery is easier, because here you have to deliver only one RNA, but here RNA plus the protein. Okay. So I will stop here and summarize what I have said. So knockout is just like a master stroke, and that results in complete and permanent loss of gene function. But knockdown is most of the time is incomplete and non-permanent activity. Okay, and the outcome of knockdown depends on the relative ratio of siRNA and the concentration of mRNA. But in some cases, knockout is ineffective. Like if you have lethal genes or genes for which that can be compensated, knockout is ineffective. In that case, knockdown gives rise to your desired phenomena. Okay, and the delivery part we have already mentioned. Delivery of RNAi is easier, but delivery of knockout, that is Cas9 plus sgRNA, is a bit difficult. So I will stop here and handle any question if you have.
How can you judge that which knockdown or knockout is effective for which gene before going to the lab?
See, there are always some bioinformatic tools available.
Always there, sir,
But the point is, right, unless you do the experiment, you are never sure whether the bioinformatic prediction is right or wrong. Gene-wise, it is very difficult to predict. Do I make my point clear? If you want to, as I mentioned that, the delivery of knockdown is easier, but interpretation of knockdown is difficult because it has a lot of off-target effects, because the phenomena that you see is because of knockdown or something else is difficult to interpret. But this kind of difficulty, ambiguity doesn't arise in the case of knockout. See, Cas9 doesn't. Similar means what? Immunity factor. See, here the immunity factor, in the case of knockdown, all RNA factors in that case are kind of immunity factors. They can be compared to the immunity factors in plants. Okay. Because I will qualify the factor immunity, I will call it adaptive immunity. Knockdown can be considered as an adaptive immunity.
My job is to conclude, sir.
Please go ahead.
Dear students, if you don't have any more questions, we profusely thank Professor Mukherjee for delivering this wonderful lecture. And I am sure you will agree that before this lecture, apparently we used to believe that the so-called RNA interference and knockdown is sort of incomplete effect, so knockout has become the final solution. Isn't it? That's what is the commonly used to believe. But now, after going through his deliberation and seeing so many examples, so many cases, our concept has changed. It is not that the knockdown has lost its relevance, rather it has its own importance. And you have seen diverse kind of examples that still many things to be understood in case of knockdown, whereas knockouts are being told to be much straightforward, right? So thank you, sir. Thank you very much for
my pleasure.
sparing your time and enlightening us for these new developments and so many examples. It was really wonderful lecture. Thank you very much, sir. So now